All right. So we're going to go ahead and get started. I want to welcome everyone today who has joined us. We're very excited to have you here for this Tech Talk with Gabe Kwong. It is going to be really interesting. I have already spoken with him to learn about his research a little bit. But I am Leanne West, and I'm the Chief Engineer of Pediatric Technology at Georgia Tech. And this is a series that we do every quarter. So feel free to join us in the future as well. But today, I want to have Gabe introduce himself a little bit and he'll give us a little high-level overview of what he does for his research.
Great. Thank you, Leanne, for inviting me. I'm very happy to share, of course, the work that's ongoing in my lab. So for those of you who don't know me, so I'm Gabe. I'm Associate Professor of BME at Georgia Tech Emory. I've been here just over 10 years. We started in 2014. And over the last 10 years, we have focused a lot of our efforts in the lab in two major areas. One is thinking about how do we develop biosensors that can pick up cancers much earlier than ever before. And then the second half of my lab, we work on cell therapies. How do we design living medicines to do all the things that we want them to do that they may not be equipped at the moment to do? And one analogy that I like to talk about, because I spoke of Leanne already before this, that most of the things I will be discussing is actually focused on our cell therapy efforts, because you may not know, but for cell therapies, children actually are the beneficiaries of cell therapies, and they're the ones that are responding the most in clinical trials. Patients who have neuroblastoma, midline gliomas, these are very young patients. And in fact, in the field of CAR T engineering, these are the ones that are responding the best so far. So it's actually very encouraging and very happy to share what's happening in that space.
Very cool. So when we talked, you talked about the cell therapies being kind of remote controlled. And so
Yes.
maybe you can talk about that a little bit.
Yeah. Well, so just give a little bit of background, maybe the 10,000-foot level is that the way we think about medicine is it principally operates along two axes. One axis is about getting spatial information, right? So like the historical advances has been in imaging, right? So MR, CT, PET, it's fundamentally about where does the cancer reside, right? Because if you knew where it was, then, and if it's localized, you as a surgeon can go in and cut it out. And even if we were at a place, say, next to a big blood vessel where it's fairly dangerous to operate, and the surgeons may not want to take it out, you can still do very localized therapies like radiotherapy to treat them. So this is fundamentally about spatial information. Now, what's very exciting is the last 50 years is that there's an orthogonal axis, which is all about molecular information, right? So how do we figure out if we can know what the molecular differences between healthy tissue and tumor tissue, we can then design what we call targeted therapies. So these therapies are like a biologic, like an antibody or small molecule that's targeting a very specific genetic lesion. And you can also do that to treat cancer. Now, the reason why these are orthogonal axes is that when you give these type of drugs, you don't really care spatially where the tumor lies within the body. And so we think at the convergence of spatial medicine as well as these molecular intelligence medicines, that will be the future where there are remote controlled medicines where we can take the devices that physicians have already used or developed and are FDA approved to then remotely control localized cell and gene therapies. And you may ask Dr. Kwong, like, why do you want to do that? Well, it's because most drugs, we cannot tell them where in the body we want them to act. And we all know that the reason why patients have side effects, drugs are nauseating, is because they don't just act on tumor cells, they act on healthy tissues. So imagine a future that we can then say, hey, I only want this drug principally to operate and turn on at the tumor site. And when it's not in the tumor site, let's suppose it's in the liver or lung, we want it to be off. And so that's the concept of remote control. Can we spatially control and turn on, tune, boost, turn off these drugs as they are operating inside the body?
Yeah, I think that's so amazing that you can have that kind of control. So, thinking about that, how do you turn them on and off?
Yeah, so that's a great follow-up question. There are a variety of different uh, instruments and medical devices that allows us to deposit different forms of energy inside the body. And I'll give you one example, which is focused ultrasound. Now, focused ultrasound is principally a mechanical wave, right? You're sending in sound waves, you see it bounce back, and then from that you can develop imaging contrast and develop a way to visualize tissues. But you can also focus energy. And if you focus, for example, ultrasound waves, you can locally heat tissues to very mild temperatures, about 3 to 5 degrees above body temperature. So it's not systemic, it's focal. And that kind of thermal energy is sufficient with our types of cell therapies that we're developing to turn on these CAR T cells, these T cells that we've modified to target cancer. So one way is to use ultrasound as a way to remotely thermally target specific sites in the body and then harness that energy to communicate with cells that we've designed to pick it up and then do something with that information.
Okay. And so, if you were going to do these treatments to look at, say, a brain tumor, um, how many treatments does it take? Is it just you do it once and you're good, or does it take multiple times to do it?
Yeah, for CAR T cell therapies, generally there are a couple of challenges in the field. I just kind of mentioned to you, even though for a lot of solid tumors, like we largely divide tumors into liquid tumors, which are more like the white cell tumors, versus solid tumors like liver cancer or lung cancer or brain cancer. Brain cancers are the ones that are actually responding to CAR T. But the challenge is that the responses aren't durable. So within a couple of weeks, sometimes a couple of months, the patients, they do recur and the tumors start going back. So the question really for us is, how do you drive deeper responses? And so one of them actually is that there are multiple groups that are doing multiple infusions. If you had like a brain tumor and you first had a surgical resection, you can actually leave behind a catheter, and it's actually right underneath the scalp, so it's actually fairly it's not a device that's like very visible, but over the course of multiple months, you can have CAR Ts re-administered at that site. And I'll just give you one more detail. It turns out that CAR T cells have a finite lifespan in terms of how many tumor cells they can kill. Just like all of us, if we work forever and we don't sleep, we won't last very many days. CAR T cells, they need rest periods. Otherwise, they become exhausted and they stop killing. And so really the trick is you want cells to be durable, you want them to be long lasting, but you also want to give them a break. So it's a bit counterintuitive. You don't always want the drug to be on. You want to give this pulsatile treatments actually to the patients.
Okay. And so you stick the CAR T cells like next to the tumor, inside the tumor that you want to treat. Is that right? And then you stimulate it by focusing the ultrasound waves where the CAR T cells are.
Yeah, so for a patient, so CAR T is not a primary first line therapy now. So typically it's after surgical resection, so there is a cavity. And so
Hmm.
you could drip the CAR Ts directly in the cavity. There are also like the ventricular space in the deep brain. Basically, it's a cavity that you can also deliver cells there. So it's local regional, and then they can migrate to the sites. But
Okay.
generally, it's never really nowadays delivered IV, intravenous, because the problem is how do you get it from the blood into the brain? And so that's people have solved it already. We actually directly deliver it to the brain.
Okay. Very cool. And so when you do this treatment, are there any anticipated side effects that this might cause by using this method?
Yeah, well, this is exactly why we want to do remote control, because the idea is that you administer the drug where it's in its off state. And when you give this heat pulse, it's transient and it's fully reversible. So now the question that maybe I can pose rhetorically is, if you had the ability to spatially and temporally control a drug, what drug would you pick? You can now be a bit creative in terms of those drugs that are so potent that exhibited a lot of toxicity because it was delivered systemically. We can now repurpose for a local delivery, right? That would preserve its local potency, but it doesn't have all these systemic side effects because we're only producing it locally. So those are the categories of drugs that we're looking at. If it's a relatively safe drug, it probably doesn't make sense to have the CAR Ts turn on and make it locally, that you can do a co-administration systemically. So things are proteins that hyperactivate the immune system. There are proteins that would even target healthy tissues, but because it's locally delivered, it just ablates everything within its vicinity.
Yeah, so it really sounds like it's hopefully going to give the opportunity to um, be so targeted that you have very little collateral damage, if you will. Yeah, that sounds amazing.
Yeah, that's the
Because I know,
thought. Yeah,
yeah, because so many cancer therapies right now, if you do chemotherapy or radiation, it still
yeah,
affects you all over, really, and not
yeah,
just in that one area.
yeah. And to be fair, what I am describing are local regional recurrence. Certainly, there are a lot of patients that are very, very sick. They have cancer cells disseminate, all throughout the
Yeah.
body. That's a little bit trickier for like spatial control, but you can still focus on command centers like the lymph nodes. Typically, are educational centers where immune cells go in and go out, and they get educated there. So there are opportunities, but what we want to first solve is if you have refractory local recurrent cancer. So like a lot of brain cancers, glioblastoma, the neuroblastomas, the head and neck cancers. Those are the ones that we want to solve first.
Very cool. Um, but yeah, but you do think it could be useful for other cancers in different areas of the body as well.
Oh yeah, totally. 100%. Yes.
Very good. Um, and so thinking about this technology, um, how far away do you think it is from actual use in a clinic one day?
That we get asked that question a lot, and I think it is incumbent on us as researchers to really kind of map out the timeline because, a lot of us may have family, friends or friends of friends that they tell you stories like, we bought a couple of computers, hold ourselves in a garage and then, came up with an app and sold it in a year and made a couple $1 million from Facebook or whatever. The timeline for life sciences is quite long, and from
Yes.
idea inception to showing that actually works, not just at the bench, but in animal models. Usually you proceed through animal models, and then safety testing and so on and so forth. Oftentimes that timeline can be anywhere from 5 to 10 years from idea inception to actually doing a phase 1B, safety slash efficacy study. So for this particular technology, we've been working on it for about 5 years. So we do have a baby company that we started a couple of years ago that we are basically optimizing this type of cell design and going after head and neck cancer first. But whatever we design and the success of that first trial in head and neck would be directly translatable for tumors in the brain. So I think another 2 years, 2 to 3 years, we will likely be able to do the first safety studies in people.
That's amazing. I mean, that's so soon in the relative timeline of developing these types of technology. So that's really, really exciting. And so you mentioned your company is working on the cells, but is there a hardware associated with it that stimulates the cells from outside the body?
Yeah, there is. So it turns out we are not a device company, but we don't need to be a device company because there are FDA approved devices. And so if you look at, I talk a lot about focus ultrasound. Focus
Yes.
ultrasound is already FDA approved to treat certain parts of the body. One is in central tremor. I don't know if you ever see those commercials where there are patients that can't even hold a coffee cup because they have very difficulty controlling the hand steadiness. It turns out that if you just ablate the subthalamic nucleus, these patients in real time, you can see them go from a shaky hand to a very steady hand, and they can hold a cup of coffee. So that type of device is a focused ultrasound device that ablates tissues by raising the temperature above, around 50 to 55 degrees centigrade. So we don't need to do that. We actually can just dial down the power setting and just do a very mild localized heating. So those devices are available Similar ones are available for prostate, for uterine fibroids. So there are a bunch of sites that you can actually target now with pre-existing devices.
Okay, well, that definitely makes your job a little bit easier not to have to do the hardware as well.
Yeah,
And so you talked about the current hardware does ablation, but you turn it down so you're not actually ablating any cells. You're just giving it that rise in heat to make the cells that you've programmed work
yeah,
like they're supposed to.
that's correct. And it's worth noting, sometimes we have people ask us, like, why can't you just ablate the tissues, like ablate the tumors? And it's because it's not curative. The tumors come back. And so we really think immunotherapy is the way to go because for these, rare case studies where patients respond to therapy, they're lifelong responders. Some patients are decades in and they're cancer free. So very similar to you get a vaccine, and if it's done properly, you could have lifelong immunity. That's also one potential advantage of doing an immunotherapy approach versus just strictly ablation.
Yeah, that's very exciting. Um, all right, so, in talking about all of this, what made you want to get into this type of research?
Well, let me just say, like, I'm sure a lot of us at Georgia Tech, Emory, and Atlanta, like, we're all basically nerds. And when I was an undergrad at Berkeley, I really didn't know what I wanted to do, but I did know I was still very interested in math. I was very interested in physics. I took chemistry. I took molecular cell biology. And by the time year two came along and I had to stitch it together to make a major, I'm like, the only thing that fit was bioengineering. So I did bioengineering. But in particular immunology, during this survey of courses as an undergrad, I took, neuroscience and, molecular cell biology. I took immunology. What was so striking about immunology to me, even then, as a 20-year-old, is that it was the study of immunology is a translational science. It's a study of how do you protect the body from viruses and bacteria and infection and so on and so forth. Whereas if you study neurons, you're studying things that are more fundamental, right? The calcium channels and action potential. So it's not really directly related to developing better ways to protect the body. So I was just very much, I guess I didn't have to do very much work to find what I really enjoy because that class really spoke to me. And from then it was pretty much developing experience in the space and then really checking off the different signs that yes, this is probably the thing that would drive me for a lifetime, and it has.
Yeah, that's very fun. I mean, it's always great when you can figure out something that you're good at and you have passion about, right? That
Yeah,
is fun and interesting to you as you continue to move forward in your career. So I love that.
yeah,
And so you mentioned having a company that you just started, and I think you've told me before that you maybe even have other companies that you've started before. So maybe can you talk about them a little bit? What all companies have you started?
yeah. I have two companies, and I just incorporated a third one yesterday.
Oh,
So
yesterday.
it's
Good.
a really infant company. But as a biomedical engineer, we are not everything that we develop has the potential to impact patient health and things like that. But I think there are moments if there are like 10 technologies that you're developing, there could be one or two that you kind of know that maybe there's a potential there. So these companies are ideas that we've all incepted either in my lab here at Georgia Tech, and one company was formed right after I left MIT as a postdoc, where the postdoc work basically gave rise to the portfolio of IP that went into the company. And all these are all focused on slightly different areas. The first company is basically sensing. How do we essentially replace a biopsy with something that can give you the same information, but potentially could be even more predictive because the biopsy is more or less a very static snapshot of tissue. Whereas if you measure biological activity or tissue activity, that's more predictive. It can tell you trajectory. And so the first company was based on building sensors that can give you that type of information. A second company is the one I just mentioned, like cell therapy, where we want to usher in an era where we call it spatial medicine. that we design cell and gene therapies and other types of therapies that have the potential to be locally turned on and boosted at sites within the body that we want them to be on. And then the third one, I'm not yet ready to share, but it's also technology that's been incubating in my lab for a number of years. So it's not something that we just, I don't really think that there's such thing as a eureka moment. It's a very long ramp up time, and you learn a lot about how to solve um, uh, the drug design and how it works in mice and so on and so forth before you're basically ready to push it out for the next level.
Absolutely. And I know, from experience, I mean, it helps to really understand the problem you're trying to treat, right? And then what's the best way to do it? And with all of your research, you end up learning along the way what works and what doesn't. So I think that's really,
Yeah.
really exciting that you've been able to start all of these companies. And I know a lot of times people who join our Tech Talk are interested in starting companies, or maybe they have already started their own companies. So I wondered if we could get a little insight from you on what it's like to be an academic researcher starting a company.
It's really fun. I mean, like I have a big sandbox, which is my lab, and then there's another sandbox, which is, working with very talented people in industry, from operational talent to executional talent, right at the bench, and there's regulatory talent, right? You might think that's
Yep.
dry, but it's actually quite very important to position the drug and the precedents in a way that it makes a lot of sense to the FDA to guide it in certain paths. I would just say, if we do our education right, which Georgia Tech and Emory, I think, is really at the forefront of education, we really want to just instill in people a sense of curiosity and problem solving. And we will never be able to teach you, for example, in our class that whatever future product that you're going to work on, this is the path forward. We can't teach you that. But what we can teach you is to make you aware that you have these analytical muscles in your brain that you may not know exist. But if you are deliberate and analytical and never satisfied with an answer, right? To dig deeper, like it's very natural to kind of take those same skill sets and move it into a more industrial setting where you can then push forward like different aspects of the company, if you want to devote that much time forward. So I would say it's very, very similar to what we do on a day-to-day basis. It is different on the outside, but the fundamental skill set is relatively the same.
Yes. And so if anyone has a question, we have about 5 minutes left. So I would love for you to post questions in the chat. Um, but while we wait on some questions to come up in the chat, I'm going to ask you another question. So, so now you've done this. Are there any tips you'd like to share about starting a company? Is there anything that you've done and you're like, gosh, I wish I had known that previously before I ran into it?
Well, one tip is like don't start a company too early if you're an academic member, partially because there are a lot of costs you have to start thinking about, right? So like even if you're to license IP from either your IP from the university or someone else's IP, you need to start paying lawyers and stuff like that. So you need to be prepared that there's bills to pay, basically. Whereas in an academic setting, if you can incubate it for as long as possible, right, to de-risk and really map out what the use case is. I think the challenge is that oftentimes the academic interest in like solving problem A, B, and C, it does not necessarily overlap with what the market need is for that space. So the quicker you align those, that there is a market need and there's something interesting that you could do in the lab, the better it is, because you don't like you don't double your effort basically. So I would say be patient and pick your right moment. You always want to think about trajectory and not necessarily the absolute value where you are. You want to think about what is my slope and start a company when you have the sharpest slope.
Very good advice. And we do have a question in the chat from Shibley, and it says, "What are the current roadblocks of scaling CAR T therapies?" So that's a great question. How scalable are these therapies?
Well, so my old PI, tongue in cheek, would say these are personalized therapies on steroids. And in a lot of respects, I think he is insightful because the issue is that the traditional pipeline is that a patient would donate their own cells. Those would be modified, those would be expanded, and then those would be transferred back. And so there are very few, tertiary hospital settings that would be able to handle the complexity of this kind of pipeline. But having said that, we are basically at the beginning of cell therapies in every single industry, right? Like the automobile industry, DNA sequencing, everything started off complex, costly, and the more demand and the more people, the more big brains start to think about solving the problem, you see these precipitous declines in like cost and manufacturing time and so on and so forth. So I 100% believe in the next 5 to 10 years, you will see very, very similar activities. My lab actually is part of, many other labs across the country that are designing new ways of manufacturing cells that are scalable, that would cost at least 10 to 20-fold less than what the current therapies are. And so it's on the horizon. So it's going to arrive fairly soon. But for right now, yes, it is a problem for scale of manufacturing.
Okay, and I'm not sure that I realized that you take the patient's own cells and engineer them and then put them back in. So that's really interesting.
Yeah.
Um, so we only have 2 minutes left. Since we don't have another question in the chat right now, I'm just going to ask you, is there anything else that you want to share with the audience that you really want them to remember from today or to take forward with them?
Yeah, I'm not entirely sure, who is in the audience. It kind of depends on, what, stage of the career is. But I guess one general thing, maybe just to reflect on what you said about, it'll be, uh, for lack of a better word, like being passionate or being devoted to a certain thing. I think it's a very natural thing, at least for me. Sometimes I have undergrads that come into my office. They say like, Dr. Kwong, like, how do you have all these things happening at the same time? And I said, well, if you like woodworking, if you like working on your automobile, like you could spend a whole Saturday afternoon working on your car and then you won't know like that 14, 16 hours have passed. And I am very, very lucky to be in a position where I really enjoy what I do, and it brings me deep contentment. So that combination is, if you are looking for something to do in life, I would say, as many people have said before, like you really want to find something where it's more like a hobby. And if it's a hobby, you won't really complain about spending a lot of time on it.
That's awesome. I totally agree. I love
Yeah.
working in pediatrics. It's so much fun to work with kids. So I think that wraps it up for today. I'm so happy that you were able to join us and share what you do with everyone. I think it's really exciting and promising, and I love that it's maybe only a few years out. So that's amazing. So thank you again for joining us, and hopefully others will join us for our next Pediatric Tech Talk. I don't know who the person is we're bringing on right now, but I'm sure it'll be just as good as today. So thanks everybody for joining.
Thank you for having me.
Yeah, absolutely.